Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 54, and it's going live on April the 10th. A wet April the 10th at that. I hope if you had a break over Easter that it was nice and relaxing. It is definitely the other time of the year, other than Christmas, when no one gets in touch with press releases or guest opportunities. For something to do, I checked out how many press releases I'd normally get in a week, and I was surprised to find 3 weeks ago, at random, the total was 573. I got AI to do that, I didn't count them all myself. This past week, with the holiday, it was 146. Now, you'd think that that's still a lot, which is true, but of those, most of them aren't relevant. Probably the least relevant this week was from a trade union about a strike at a metalworks. Last week, we had a conversation with Manish Arora, CEO of Linus Bio, on autism, and we chatted with Thomas Klidal, CEO of Synucleino, about improving kidney transplantation outcomes through better cytomegalovirus prevention. This week, because April the 11th is Parkinson's Disease Day, we chat with Dieter Willbold, CSO of Priavoid, and Antje Willuweit, Director Preclinical Development of Priavoid, and our other interview is with Karsten Eastman, CEO and co-founder of Sethera Therapeutics. And that conversation is about a new way to build more stable and drug-like peptides. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, or sponsoring a podcast episode, or having us cover an event. Because we didn't have the same number of articles this week as we normally would, we will skip the roundup of news over at deeptechdigest.com. But please don't let that stop you from heading over there, because there are still some very interesting articles this week, I promise. All right, let's kick things off with peptides. To talk about a new way to build more stable and drug-like peptides, which could potentially transform the industry, we have a conversation with Karsten Eastman, CEO and co-founder of Sethera Therapeutics. Hi Karsten, thanks for braving the time zone difference this morning. Maybe we could start with some information about your company and also about your work with the University of Utah.
Sure. The company is Sethera Therapeutics, and we're actually a spin-out from the University of Utah. The work was, luckily enough for me, part of my graduate career. So I found an interesting enzyme initially, and we did some work with it. And we found that the enzyme has an unusual property of controllable promiscuity, which is a very unusual thing to see in the enzyme space, meaning that we can use one enzyme for a lot of different work, but it's not like it's going haywire the whole time. It can actually allow us to do some really unique things, but in a controllable way. And so we've been building. We are operating really out of two locations in downtown Salt Lake City at the Altitude Incubator space. And then we're also leasing space from the University of Utah to really help get Sethera started and move this forward. So it's been nice to remain so close and connected to the university still.
You mentioned that you're based in Salt Lake City and at the university. Is there, everybody thinks of Boston and California as the hubs. Is there much of a life sciences hub in Utah?
So I think that the life science scene in Utah is growing significantly. The Altitude Incubator has been pulling a lot of unique people and unique talent. The University of Utah has a focus right now to spin more companies out, and they have a lot of work on the, say, bench to bedside type of programs. They also have programs that they're trying to build tech that enables remote and austere conditions where new technology can be deployed in those unusual circumstances. So I I think that while Utah's been known primarily as sort of a med tech hub, the biotech space is growing at a really nice pace.
And I guess in Salt Lake City and around Utah, as opposed to some of the other major life sciences and health tech hubs around the country, you have more room to grow, I guess. And also you have the university as a hub in of itself with people coming out of there, also with spin outs coming out of the university.
There's lots of space to grow here, and I think that there's lots of opportunities. That one of the neat things is the people that I've hired for Sethera moved to Utah, and they've really enjoyed living here so far. So that's been nice to see.
Yeah, definitely. Coming on to peptide drugs, what are the current issues with those?
Sure. I think that the easiest thing in terms of thinking about issues with peptide drugs is they have a very short half-life in most scenarios. Peptide is just a small protein. They're both chains of amino acids. And one thing that the body is exceptional at doing is recycling things. And one of the things that are recycled are amino acids from peptides. So whenever you say eat a protein, it breaks it down to individual amino acids. Similarly to whenever you take a peptide, your body will say take this bioactive form and then shorten it and shorten it. And sometimes even one proteolysis event, which is where it's chopped or cleaved, is enough to render the peptide completely ineffective in biological contexts. So the biggest problem that I see is there's very short half-life.
And so, what's PAP-B and how does that work?
So, as I mentioned earlier, we put to work a very interesting enzyme, and that enzyme is called PAP-B. In nature, what it does is it forms six crosslinks. So you can think of a peptide kind of like a noodle. It will flop around, it will change, it will twist, it will contort. What a crosslink does is lock that peptide in a specific conformation. So when you read about peptide therapeutics online, one common thing you'll see is a macrocycle, which basically just means the peptide is cyclized. What PAP-B does is it's an enzyme that actually installs six crosslinks on its native peptide substrate. And we thought that that was really interesting and started to find ways to use the enzyme to not just go after what it transforms in nature, but be able to transform many different peptides. The reason why this is so important is, well, structure is function. That's biology 101. And in nature, there are a lot of unique structures that enable different types of biological interactions. So, say insulin, that's a peptide. Insulin has a very unique architecture. It has two different chains, and the chains are linked together in several ways. Most peptide therapeutics are very limited in the types of architectures that they take. And one of the reasons is that they cannot screen effectively these higher order architectures. They can say screen a linear peptide, they could screen a macrocycle, and then some people are able to do these constrained bicycles that look a little bit like a heart. And that's great. And that's produced some really interesting structures and interesting therapeutics. And that macrocyclization process actually helps increase the half-life that I mentioned earlier, but it's still very limiting. You could have all of this chemical diversity that you want on the outside of your macrocycle, but if the only thing that you're making is a circle, you're inherently limited in terms of what you can do in drug discovery.
I've heard or seen the claims about what you're working on being able to transform drug discovery. How could you maybe explain a bit about that?
Sure. One of the things that's necessary when you're working with, say, an established target. So this is a biological target in the body that you either want to find something that binds to, find something to activate it and make it do work, or find something to, say, deactivate it and make it stop. That process often starts with screening. And you could do, say, physical screening where you actually have all of your compounds in real space and time, or you can do in silico screening where your compounds are all basically digitized and you do your screens in silico, so on a computer. And a lot of the time, you're going to be doing both as you're finding your first hit, and then you do hit to lead, and then you do lead optimization. What we've spent most of our initial time focused on is making the most diverse libraries for that initial screen in real space and time. So rather than being limited to linear peptides or just a simple macrocycle, what we've used our platform to do is build diverse architectures in massive libraries. And this is how we believe it will transform drug discovery. Rather than being limited to those few cases, we can make very intricate and interesting peptide structures and then vary all of the chemistry of those peptide structures. And that's how we think it will transform it. Rather than say, here's your biological target, here's your macrocycle, it can hit one location here. We can chain together macrocycles or have multiple unique architectures that can make very unique interactions with biology.
And is that going to help with what's previously been considered to be undruggable targets?
Absolutely. And the reason for that is one of the easiest ways to think about an undruggable target is, say, their protein-protein interactions in many cases. There's other undruggable targets, but that's the simplest one to think about, where you have a protein here and a protein here, and you have this large interface between the two proteins. Now, small molecules by their name and definition, are inherently limited in space and size. And small molecules cannot disrupt, like, they cannot disrupt this protein-protein interaction because they're too small. It'll still come together. What peptides and higher order architecture peptides can do is basically displace or shift or interrupt that interface. And then all of a sudden you can block this previously undruggable protein-protein interaction. And that's why it comes down to a space and size and how it interacts with a target.
And so from there, what are the potential applications for this?
The potential applications, I think this is both an fortunate and unfortunate thing where it's truly target agnostic and indication agnostic. Realistically, if there's a biological interaction that takes place, if you have the right architecture and the right chemistry, you can block, activate, or deactivate that interaction. So the potential applications are functionally unlimited as far as I can see.
And so, with regards to those applications, are you already in discussions about some of those potential uses?
Yes. And this is where having a new technology like this is a neat double-edged sword. A lot of the discussions that we've had, people have said that, they view us as a true new modality, meaning they can't put us in the small molecule box, they can't put us in the normal peptide box, and they can't put us in the biological box, like, say, antibodies. And one of the biggest questions that we've been asked is, well, what is it going to look like once your peptides are in animals? Which is a great question, and that's something that we've been actively building towards, because since what we're doing is so different, they want to ensure that they have reasonable data and examples of the peptides being safe, effective, well tolerated, and I guess functionally useful. So while we've been building the screening platform first, we now are saying, let's take some of the molecules from those screens and start moving them towards the clinic.
And so, are there other things that you're working on with your platform?
Yes, and I can't get into too many details there, but we do also recognize that there is a direct way of using the platform in diagnostics and a few other select uses that are outside of the therapeutic space as well.
And I appreciate that you can't talk about necessarily everything that you're working on, but do you already have any partnerships in place with positive results from that that you're able to share?
Sure. We do have some partnerships in place and that we look forward to being able to announce them in the near future. But what I can say is that we've done screens on 7 different, very diverse targets so far and seeing some very promising initial results. And these targets span everything from oncology to anti-infective targets, as well as anti-inflammatory as well. So we're really trying to go after some broad, unique, and diverse targets while we actively push the programs forward.
What do you think that the future holds for peptide drugs? You mentioned some of the challenges earlier, but some of the things that you're working on?
I think peptide drugs will end up being the preferred platform for individualized medicine. One of the really neat pieces is nature has already demonstrated to us that peptides, as a therapeutic, can actually discriminate between different receptors and receptor isoforms. So meaning you can have a family of receptors that are all very, very similar, but peptides can find the right way to hone in on just one member of that family, or say 2 members of that family. A great example of that is octreotide, which is commonly used to treat the symptoms of various cancers. And it is quite adept at discriminating between the different receptors in the somatostatin receptor class. I think that that will be pushed further, and eventually we will be able to make peptides that are tailored for specific individuals or say specific families that contain even point mutations in a receptor. So I think that really the future is very bright for peptides because they can be so specific.
Is AI affecting that in any way? Is that accelerating things or making any changes?
One of the pieces that I think is necessary to really bolster AI in the peptide space is more target interactions that can directly be observed. AI is great when you have millions, billions, trillions of data points showing what is a positive interaction, what is not an interaction, and what just, is nonsense. But the unfortunate piece is there's limited amounts of data on peptides interacting with receptors and peptides interacting with biology. It's not that that data necessarily doesn't exist, it's just that there's not enough of it to have a really robust AI platform. And that's actually where us at Sethera, we are upstream of AI, meaning that we can generate and do generate all of the data that's necessary to start building those more robust AI and ML models.
And so, what does the future look like for the company? What kind of areas do you see yourself moving into?
The biggest piece that we're working on right now is taking a candidate from one of our screens towards a clinical developmental path. We want to be a preferred screening partner for a lot of big pharma because the space that we think is our biggest strength is just how robust the screens are. But we recognize that until we say take a Sethera molecule and put it in an animal and then start taking it towards a clinic, there's going to be some hesitancy about using us as a screening partner. So that's the current path that we're on. I'd say we've had a very robust platform in terms of the screen. Now we're going to show that our peptides and molecules work in the clinic.
How do you bridge that gap between actually making that a reality?
We've taken some members from the screens and we moved them into cell tests. We're now moving them into mouse models, and depending on how the mouse models go, start moving them towards non-human primates, get a nice amount of efficacy, safety, tox data, and then start building a reasonable case for an IND application.
All right, sounds like you've got the future mapped out anyway. Does the clinical trial pathway look like? Is that you doing your own clinical trials or is it partnering with companies on their clinical trials?
I think it will depend on the use case and the indication, and if we come into contact with the right people at the right time. There is a scenario where we do our own clinical trial, and I think that some of the molecules that we are interested in, there will be definitely an appetite for them. Or if we come across the right big pharma partner that is interested in one of the indications that we've already screened, or has their own indication that they would like screened, that's where someone else could say be the partner that takes our molecules forward into those clinical trials.
Is there anything that you wanted to talk about that we haven't covered?
I think that we covered everything really nicely. I appreciated the chance to say the pieces about the individualized medicine and how there is a really robust future for peptides in AI and ML. And I just really appreciate the opportunity to chat with you about this.
Are there other companies doing anything similar to what you're doing?
There's a few you could look up right now. So there's a PeptiDream. Based in Japan. They have a peptide screening platform that's been routinely used. They have a really neat way of using unnatural amino acids to do their cyclization, and they've really built a robust screening platform and have been used by big pharma quite a bit. There's also Bicycle Therapeutics. They're actually UK based. They started to move away from being a screening partner and started developing their own therapeutics in-house, and they've been mainly focused on oncological targets. Then there's also Unnatural Products, who just had a fairly large deal with Merck. And they also have a peptide platform based on macrocyclization where they're really focused on orally bioavailable peptides. So that's where they're trying to set themselves apart. And in each of those cases, the peptides that they make it's really difficult to control the chemistry of multiple crosslinks. And that's why most of these platforms have limited themselves to, just the single macrocycle or the constrained bicycle, like I mentioned. Controllably having more crosslinks than that is a very difficult thing to do.
I assume that there's plenty of space in that sector of the industry for everybody to be able to do well in that.
I think that there is reasonable space, and I think that there's a lot of opportunity for each of the individual platforms to make their own mark. I think that our goal really is to demonstrate that it's necessary to have this higher order architecture to make a good therapeutic for certain types of indications.
Another company definitely to keep an eye on. Next, tomorrow is Parkinson's Awareness Day, and it's significant for me personally, as my mother had Parkinson's. One company doing all it can in the space is Priavoid. So, time to welcome Dieter Willbold, CSO, and Antje Willuweit, Director of Preclinical Development at Priavoid. So thanks both for joining us today, and I wonder if first you could tell us about the origins of the company.
In an academic setting, we have developed a unique mode of action that we have realized first for A beta as a target protein. And the compound that came out of it is called Pre-002. And went in the academic setting until a phase 1 study even. But then we finally had to found Priavoid in order to bring the compound and the mode of action through further clinical development stages, and in parallel to transfer this mode of action to other target proteins like α-synuclein, and their Pre-101 came out.
So could you tell me a little more about Pre-101 and how that works?
The mode of action is actually based on the observation that the monomer of α-synuclein is stable, but only metastable in our brains. And there is obviously a more stable conformation, which is in aggregates, in fibrils, which is containing the chemically identical building block as alpha-synuclein monomers that fulfill their function at presynapses. And this is very obvious because otherwise aggregation would not happen. So the aggregated conformation is more stable than the conformation of the functional alpha-synuclein monomer. And our mode of action is to stabilize the monomer that far that it becomes the most stable conformation, and this automatically disassembles toxic aggregates into harmless and possibly even functional monomers. And this is the mode of action that we realize for various target proteins. And in this case, it's Pre-101 with α-synuclein as a target protein.
Do you expect that this could be perhaps a transformational drug for Parkinson's disease?
Yeah, so Parkinson's disease, of course, is the second most worldwide neurodegenerative disease, and it's, of course, with a very high medical need. And we think that with our compound that we developed, we could achieve disease-modifying therapy. And yes, we think that should have an impact. If it's working, of course, that we don't know yet.
And what effects do you anticipate that it will have for patients?
We want to disassemble these toxic aggregates, which are in the brains of the Parkinson's patients and lead to these well-known motor symptoms. And we want to disassemble these toxic aggregates. So we will eliminate the toxic species out of the brain from the patients. And that should slow down the disease progression at least, or maybe even stop the disease. And of course, lost neuron, a dead neuron, cannot be brought back. But there is a certain reserve of the brain, and there might be neurons which are damaged. They lost their synapses and their contacts. And if we remove the toxic aggregate, then the neuron might bring these synapses back, so that there might be even that like in a benefit. But this we don't know. We aim at least stopping the progression of the disease, that the patient, once it's diagnosed with Parkinson's and takes the medication, will freeze that status at least.
Do you think that there's potential to reverse some of the effects of the disease? I know you mentioned that neurons can't be brought back.
Yes, some synapses can maybe grow back, and that would also reverse symptoms. But this is really hard to tell from the status of the program at the moment. And we aim for, in principle, for a monotherapy. But of course, there are still the other symptomatic treatments which are now on the market. They're still available.
Do you anticipate that it might be something that can be a manageable disease?
Yeah, I think what is really crucial is an early diagnosis, because if you diagnose it very early, then of course there's not much damage. And there's also really, in the last years, there was really some progress in the diagnosis of the disease, also of the disease cause. So now we are able to detect these aggregate particles in the CSF, so the cerebral spinal fluid, and also the blood, and can have a diagnosis of Parkinson's or Parkinson's-related diseases much earlier. And the earlier we know that the person is on that track to Parkinson's, the earlier we can treat. And then that would really have then an impact on the patient's lives.
And we're talking about Parkinson's disease, but is Pre-101 applicable to any other conditions?
There are other synucleinopathies, which is, for example, MSA, so multiple system atrophy, and also DLB, dementia with Lewy bodies. And they are also based on α-synuclein aggregates. And our compound can also target those exactly in the same way. So because it's binding to the monomer in the aggregate or in the fibril, we can also target the aggregates formed in MSA and DLB patients. And we also anticipate that our drug would be beneficial for those.
And is it used as a standalone treatment, or have you investigated its use in combination with other treatments?
So not yet, because we are at the very beginning of the development, but we think it will be a monotherapy, but of course a symptomatic treatment in addition would be possible if needed.
Of course, we have to go for a monotherapy for the development of the Pre-101 compound. It's just not really feasible to start with combination therapies from the beginning on. And of course, all therapies that will somewhen be on the market, whether they can be combined, this is something we cannot know at the moment.
So could you tell me what about the clinical trials that you are working on currently?
Right. So PRE-101 is in the preclinical phase. So we have just finished the preclinical data package. That means we have shown efficacy in animal models. And we are, of course, planning going forward to do clinical trials. But so far with this program, we have not achieved that. But with the other compound, which is against Alzheimer's, there we have a clinical trial running. And Dieter can maybe say a few more words about that.
The compound PRE-002 that we have developed to disaggregate A-beta aggregates into harmless monomers. This compound is now in a clinical phase 2 study, and actually it's quite far already. So next week, the last patient will get the last dosage of the trials with a follow-up period and further database cleaning and so on. We should have results by the end of the year. But the mode of action is exactly the same, stabilizing the monomer, in that case, the A-beta monomers, and thereby disassembling, detangling toxic A-beta aggregates into harmless monomers.
When it comes to the design of the clinical trials, how important is the stage at which the patients are? Clearly, Parkinson's has that range from you can start to see the signs right through to extremely severe symptoms.
Yeah. So in principle, our mode of action is designed that it would help or disassemble the aggregates regardless of the stage. So either in the early stage or in the later stage. But for designing a clinical trial, you would normally go to early stages because the driving force of the disease is clearly done by α-synuclein aggregates and not secondary inflammation or whatever would damage neurons in addition. And therefore, for really showing proof of concept, one would design the trials very early with patients with very early stage in order to see the best efficacy.
And you recently gave a presentation in Copenhagen. Could you tell me about that and what the reaction has been like to presentations?
Right. So Dieter gave a presentation of PRE-002 against Alzheimer's, and my colleague Mark Sevenich had a poster presentation on PRE-101 for Parkinson's. And this was really a very positive reaction we received from as many interest from academia and also industry. He was really standing and presenting at his poster many hours and had no rest. But we were very lucky that we had so much good feedback. And actually, we also had 2 more posters on earlier programs with the same mode of action, but also different diseases, different targets. So we had a bit like a roadshow on the last ADPD conference in Copenhagen.
And so what are the next steps for PRE-101?
Yeah, so we are at the moment starting to plan so-called IND-enabling studies. So these are all toxicity studies, for example, toxicokinetic studies, which are mandatory for developing a compound further into clinical development. And this is the aim, what be done with PRE-101.
And so what else is in the pipeline? You mentioned 102. Is there anything else that you're working on currently?
Right. We have a second program on Alzheimer's, which is based on tauopathy, so tau as a target. And we also do have a program on ALS with TDP-43 as a target and also some undisclosed targets.
How important are partnerships and fundraising to the company?
Very important, I think, for the clinical candidate PRE-002. This is really mandatory. Maybe also Dieter can say a few words. Of course, the next step would be a clinical phase 3, which really costs a lot of money.
Yeah, and even the phase 2 cost a lot of money, more than 60 million euros. This funding we have received from the German government, from an agency which is called Agency for Disruptive Innovation. And the idea was, of course, to check whether this unique mode of action of stabilizing monomers can really do beneficial things for patients. And if yes, in the case here, in the phase 2 study of Alzheimer's patients, the concept is valid, should be valid at least for other neurodegenerative diseases.
Has the introduction of things like artificial intelligence, machine learning, have they been helpful in reducing any of the costs of trials?
Not yet. I think in our case, with our compound, the AI is not powerful enough, but I think it will be more in the future. I think it is really having increasing power from year to year, and we expect, of course, that it will have an impact in our future drug development.
How important are awareness days and awareness months in advancing the cause of treatments?
I think it helps, of course, to have this media to be aware of things, of a disease, and then, of course, also of companies trying to develop treatment against it. Of course, it helps, especially small companies like ours, to be presented there. But I would also say that everybody knows Parkinson's, right? But diseases like MSA, for example, are less known. And it would also help to present more on these orphan and rare diseases.
Is there anything that you wanted to talk about that we didn't talk about with regards to the treatments that you're working on?
I think one aspect that we have mentioned, how far the disease should be or can be progressed in a clinical trial, you need to consider that the patients need to be able to give informed consent of the treatment in such a study. And that limits, of course, the late disease stages, because usually the patients are not able anymore to give consent, informed consent, to take part in such a study. So this is one of the most important reasons why really late stage diseases are mostly not represented in clinical trials. Maybe MSA is one exception, only because MSA is really progressing really fast, much faster than Parkinson's disease.
Are there a lot of companies working in, obviously there are a lot of companies working on things like Parkinson's and Alzheimer's, but are there any that are working on any similar treatments to the ones that you're developing?
We think really the reversal of aggregation is something unique, but there are, of course, a number of companies that claim to work like this with their compounds, and companies that focus on inhibition of aggregation with their compounds. And of course, the companies that develop antibodies against α-synuclein aggregates are also present in, of course, the ADPT conference in Copenhagen, and also in development of drugs, of course. We have really the information already now from our phase 2 study that the safety is really great. So safety and tolerability of our compound is really great, and in comparison to competitor drugs. And this has several reasons. First of all, our compound is by definition a small molecule, but it's a large small molecule. It's just by definition a small molecule, and because our compound is larger as an all-D peptide, our compound is more specific, has less off-target hits, and therefore less adverse events, less side effects, and also when degraded in the body, our all-D peptide, if they are degraded at all, are degraded into, for example, D-amino acids residues or T-amino acids, and these are not toxic. They are actually, we do have enzymes that convert them into L-amino acid residues, and these L-amino acid residues go their normal anabolic or metabolic pathway without being toxic metabolites. So these are all reasons and explanations why we observe a really great safety and tolerability profile for our compounds. And also in comparison to antibodies, where you certainly know that the anti-A-beta antibodies that are on the market do cause side effects like amyloid-related imaging abnormalities, ARIAs, abbreviated, and these are really based on their properties as an antibody. And because our compounds are not antibodies, we never observed such ARIA side effects with our compounds.
for 3101, which is directed against α-synuclein, which is an intracellular target. So that means the compound needs to go into the cell in order to disaggregate the fibrils. And this is really hardly possible by applying an antibody. But our compound is able to go into the cell and to act there where the aggregate is located.
Let's hope that Parkinson's can be beaten in the future. As I mentioned, I've seen its effects firsthand, and it's a terrible disease. And let's hope that Priavoid and others in the space can be a part of a Parkinson's disease-free world in the not-too-distant future. So that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.